Welcome to Quantum Rabbit, where we make videos of mathematics, physics, chemistry, and biology. In this video, we discuss the pathophysiology of SARS coronavirus 2. Part 1. Genes and Proteins. In this image, we see the SARS coronavirus 2 virion, which contains a lipid bilayer that forms a vesicle. Within the vesicle, genetic information is found in a single segment of positive RNA. This information is compacted and protected by nucleocapsid proteins that bind to RNA, which together form a polymer. The lipid bilayer contains 3 membrane proteins. The M, or membrane protein, gives it stability and binds the genome by joining the nucleocapsid proteins. Protein E, or envelope, is a useful ion porin for regulating osmotic pressure. And the S, or spike protein, serves to bind to cell proteins and promote membrane fusion to enter the cell. The coronavirus genomes are the largest among the RNA-positive viruses, with a size of 29,900 nucleotides and containing 10 genes in which the information encoding 30 proteins is found. The first gene encodes a polyprotein that can be synthesized in its full or short form. This polyprotein is cut into 16 proteins that we call non-structural proteins, or NSP. The second gene is the one that codes for spikes. The third gene codes for accessory protein 3A and contains 3 other overlapping genes that code for accessory proteins 3B, 3C, and 3D. The fourth gene codes for protein E. The fifth gene codes for the M protein. The sixth gene codes for accessory protein 6. The seventh gene codes for accessory protein 7A and contains an overlapping gene that codes for accessory protein 7B. The eighth gene codes for accessory protein 8. The ninth gene codes for protein N, but contains 2 overlapping genes that code for accessory proteins 9B and 9C. And lastly, the 10th gene codes for accessory protein 10. These NSP and accessory proteins are not part of the virion, but some are used to evade and inhibit the immune system. Others modify the cellular metabolism to benefit the production of viral proteins. Others modify the endoplasmic reticulum to form replication centers, and others to replicate the viral genome. The virion is the infectious form of the coronavirus. It seems to just be a bubble with 4 proteins and a nucleotide polymer with no life of its own. But make no mistake, the shape of a virion is not its only identity. Once it manages to infect a cell, the virus's metabolism is activated and it comes to life within the cell. The virus in its active form begins to metabolically and morphologically modify the cell to favor the production of replication centers and the synthesis of viral proteins. Not only that, it can also inhibit the signaling of the immune system and apoptosis, giving time for hundreds of virions to be generated that are released with the purpose of finding other cells. This concludes our discussion on the genome and proteins of the coronavirus. Please remember to subscribe and like the video if you would like to see a part 2. And leave a comment with any feedback or video suggestions. In the next chapter, we will see the immune system in the nasal mucosa versus coronavirus 2. Who will win?